As of March 1st, 2022, there have been over 363 million COVID tests using qPCR technology in the US alone. This is not the only application of qPCR, however, as it is the golden standard of diagnostic testing for viral and bacterial infections as well as for genomic testing. These machines are definitely already available, but the main problem is cost. They average anywhere from $10,000 to $40,000, with the even more advanced machines costing upwards of $100,000. This is the problem the Quantitative Cytokine Detector, or QCD, aims to solve. From the traditional 96-well plate that is often used in PCR, we cut this plate down to just 5 wells, with these 5 wells being able to produce the same valid and precise results as normal qPCR. This is achieved partly through the use of the commercially available Pocket PCR, a handheld and open-source thermocycler. This is paired with a custom-designed spectrometer, capable of being adapted to multiple wavelengths often used for different qPCR fluorophores, making it bear varied use with the biomedical diagnostic sector. The QCD has a selling price of $2,500, making it a fraction of a price of the normal qPCR machine and allowing it to be widely more applicable for individuals or smaller institutions to harness the power of qPCR without compromising on quality or validity.
I'm Garrett, the team lead for QCD. Over the past 2 years, COVID-19 has changed the rules and function of not only our Florida tech community as we know it, but also the world at large. As of March 1st, 2022, there have been over 363 million COVID tests using qPCR in just the US alone. In this time, the importance of fast and reliable diagnostic testing has been paramount, and qPCR has been able to satisfy this need and has entered the limelight as being the golden standard for testing of viral and bacterial infections. However, the main issue of qPCR has also been firmly cemented: its exclusive cost. Most qPCR machines on the market today range anywhere from $10,000 to $40,000, which leads to an exclusion of smaller research institutions or even developing countries from being able to access the diagnostic capabilities that have proved to be so essential during the course of this pandemic. This is where QCD comes in. Aimed at providing the same qPCR technology at a fraction of the cost, QCD offers a comprehensive qPCR testing platform at a price of only $2,500, a fraction of the price of a traditional qPCR machine.
To accomplish these goals, we split the project into 2 teams, a PCR team and a spectrometer team. My name is Noah, and I am the spectrometer lead. The spectrometer circuit employs a multi-laser setup, which is controlled through a steady-state relay by an Arduino Uno. This allows for easy control of which laser to operate at a given time. Since the Arduino is incapable of delivering the power required to operate the lasers itself, power is separately supplied to the lasers via a 12-volt voltage converter. From the laser diode, the light emitted is channeled into fiber optic cables by passing through a collimating lens, then a focusing lens. A reverse splitter is utilized to remove the task of constantly switching out fiber optic cables. Next, the cuvette holder is where the sample containing DNA is placed for analysis. 3D printed constructs are used to accurately position the micropipette tube in the cuvette holder for the laser to pass through. After that, the light passes through a bandpass filter, which filters out the light emitted by the laser and passes the remaining light onto the spectrometer for analysis, which will be displayed on the screen. The advantage of this setup is customization. The laser setup allows for multiple lasers to be present at once, and they can be easily swapped out for one of a different wavelength. The bandpass filter also has a cartridge system allowing for easy swapping out of filters.
I'm Devin, and I'm the PCR lead. An open-source thermocycler known as a Pocket PCR was utilized to conduct the PCR reaction in our device. The display of the Pocket PCR was emulated using Arduino libraries to allow for control of the thermocycler and customization of cycles from a laptop. The PCR process was segmented into 3 programs: initial denaturing, amplification, and final elongation. The amplification program is run as many times as necessary to achieve the desired level of DNA amplification. Measurements were taken at the conclusion of each program or every 4 cycles. For our purposes, the IL-6 gene was amplified from synthesized cDNA. cDNA was synthesized following the isolation of RNA from mouse fibroblasts. Cybergreen was selected as a fluorophore for its simplicity and versatility. However, this device can accommodate any target sequence with any fluorophore. A reaction volume of 40 microliters has shown to produce the most consistent results.
My name is Kat, and I'm the research lead and lab coordinator for this project. The combination of the PCR thermocycler and spectrometer allows us to emulate and simplify a qPCR procedure. In order for the DNA material to be in an amount large enough to detect via the spectrometer, the cDNA needs to be amplified using the PCR procedure. It's placed in the Pocket PCR thermocycler where it undergoes cycles of heating and cooling that repeatedly double the amount of target sequence of the cDNA. This PCR uses a fluorescent dye-based procedure. A laser is shot at the PCR products to excite the dye. The fluorescence dye has an emission around 490 nanometers that can be detected using a spectrometer. The absorbance values are plotted to form a curve that can indicate the concentration of the target of interest. Together, this spectrometer and thermocycler device can be used to detect the concentration of biomolecules in solutions. It makes performing and analyzing PCR cheaper and more accessible than conventional methods. At a selling price of $2,500, QCD allows users versatility with the gold standard in diagnostics without compromising on validity of results.